Hello students, welcome back to a new video of Sunanda's tutorial. Today we will discuss about the heterospory and seed habit which is related to your Pteridophyta unit. So what is heterospory in pteridophytes? Most of the pteridophytes produce one kind of spore. Such pteridophytes are known as homosporous. Homo means same and sporos means spore producing. And this phenomenon is known as homospory. However, there are some pteridophytes which produces two different types of spores, different in terms of size, structure and function. Such pteridophytes are known as heterosporous and the phenomenon is known as heterospory. The two types of spores are microspores and megaspore. Microspores are smaller in size and develop male gametophyte, while megaspores are larger in size, develop into female gametophyte. According to Rashid, in 1976, he told that there are 9 genera of pteridophytes which show heterospory and they are heterosporous, which are Selaginella number 1, number 2 Isoetes, number 3 Stylites, number 4 Marsilia, number 5 Pillularia, then Regnelidium, then Salvinia, then Azolla and Platysoma, which is the 9th one. So these are the genera which show heterospory. Then origin of heterospory. How this type of phenomenon have originated? First, paleobotanical evidences suggest that the heterospory arise due to the degeneration of some spores in a few sporangia. As more nutrition becomes available to less number of spores, the surviving spores grow better, hence increase in size. So the paleobotanical evidence told that heterospory arise due to degeneration of some spores. Inside the sporangia, some spores gets degenerated and some alive, some spores grow better with better nutrition. As some become degenerated, the remaining spores get better nutrition, so grow better and hence increase in size and they become megaspore. And the spores which get less nutrition become microspores. Then evidences from developmental studies. In heterosporous pteridophytes, the development of micro and megasporangia follows same pattern. They have identical organization but their sizes are different. While megasporangia, most of the spore mother cell or SPM from which the spores are developed. The spore mother cells degenerate, but in microsporangia only few mother cells are disorganized. So in case of megasporangia, many megaspore mother cell degenerate. So the number of megaspores are less, but the microspore mother cell are more and less disorganized. Only some of the microspore mother cells are disorganized. So their numbers are more. So they are small in size as they get less nutrition. As they are more in number, so nutrition gets divided and each one of them get small amount of nutrition and become smaller in size. But in contrast, the megaspore, the spore mother cell, many spore mother cell degenerate. So they get large amount of nutrition. As their numbers are very small, they get more nutrition and become healthier and large. Then evidences from experimental studies. Experimental studies said that the Selaginella marsilia suggests that nutritional factors mainly govern the heterospory. So by taking Selaginella and Marsilia, they concluded that the nutritional factor is the main factor which governs the heterospory. Under condition of low light intensity, low photosynthetic activity, Silaginella was retarded and it produces microsporangia in these low conditions. By sudden lowering of temperature, the size of microspore in sporocarp of Marsilia increases 6 times. So, in low light condition and low photosynthetic activity, Silaginella produces microsporangia. But by lowering the temperature, the size of microspore inside the sporocarp of Marsilia, sporocarp means the spore producing organ of Marsilia, they increase in size by 6 times. So they become megaspore. So here the many physiological factor or nutritional factor plays main role. Then biological significance of heterospory. The phenomenon of heterospory is of great biological significance on account of the following facts: that development of female gametophyte starts while the megaspore still inside megasporangium. The development of megaspore or female gamete takes place when it is still inside the megasporangium where it is developed. Same is true for microspore. They also start germinating while inside the microsporangium. The female gametophyte derives nourishment from the sporophyte, that is female gametophyte is dependent on sporophyte for their nourishment. The dependence of female gametophyte on sporophyte for its nourishment provide better starting point for development of new embryo than independent green prothallus which has manufactured its own food. So the gametophyte, female gametophyte or male gametophyte are in growing stage, so it is better to derive nutrition from the existing sporophytic plant than to produce own food. So what is seed habit then? The adoption of heterospory, that means production of two types of spore, and retention and germination of a single megaspore within megasporangium to form a female gametophyte led to the phenomenon of seed habit, a characteristic features of spermatophytes, that means gymnosperm and angiosperm. They produce seed. Gymnosperm and angiosperm produce seed, but gymnosperms and angiosperm, pteridophytes show some characteristic features which are related to those seed habit. So the number one point is heterospory. So it is a very important prerequisite of seed habit. Heterospory, retention of megaspore within megasporangium, germination of megaspore or single megaspore when it is inside the megasporangium. All these things are very related to seed habit, and seed habit which is a characteristic feature of gymnosperm and angiosperm. So in this way, this gymnosperm is related to your Pteridophyta is related to gymnosperm and angiosperm. A seed is the ovule which contain embryo developed as a result of fertilization. The origin of seed habit which is associated with the following. Seed habit, the origin of seed habit is associated with the following: production of two types of spore, reduction in the number of megaspore finally to 1 per megasporangium, retention and germination of megaspore and fertilization of egg, continued development of fertilized egg into the embryo while still in situ. The fertilized egg after fertilization, that means fusion with the male gametophyte, present or grow inside that megasporangium and develop into embryo, and it is grow in that position which is known as in situ. Then from the above observations, it is concluded that the life history of Silaginella approaches towards seed habit because of the following features. Silaginella have heterospory. Syllagenella show heterospory, but they lack seed habit, but they approach towards the seed habit because of the following features. Number 1, the occurrence of heterospory. Then number 2, germination of megaspore inside megasporangium. Third one, retention of megaspore while inside megasporangium, either till the formation of female gametophyte or even after fertilization. Then development of one megaspore per megasporangium. For example, some Syllagenella species of Syllagenella have one megaspore per megasporangium. And all these habits of Syllagenella, in the life history of Syllagenella, shows their tendency towards the production of seed habit. Even though seeds are not formed in Syllagenella, even then Syllagenella have these properties, but after then also they lack seed. Why? Because megasporangium is not surrounded by integument. They lack protective layer. Then retention of megaspore permanently inside megasporangium has not been well established. Then the embryo immediately gives rise to sporophyte without undergoing any resting period. Then absence of any histological union between megaspore and megasporangium. So these are some points for which Syllagenella failed to develop seed, and they placed in Pteridophyta. So it is all about your heterospory, which means production of two types of spore, and this phenomenon of heterospory present in some members of Pteridophyta, which shows their relation in evolution with the spermatophytes, that is gymnosperm and angiosperm. They show heterospory but lack the production of seed. They do not produce seeds, but they have some seed habits, that and they have some characteristic features which are prerequisite to the seed habit. So it is a very, very important topic from your examination point of view. It is a very important short notes, and very frequently comes in your semester examination, and as well as the name of this genera which show heterospory comes in your competitive examinations. So I hope you really enjoyed this session, and in my next video I will upload other topics related to pteridophytes. So stay tuned and enjoy my videos like this. Thank you.